logged in remotely. Can you hear me? Okay, great. So we usually start at 4:01. I don't know if it's 4:01 yet. So it is my pleasure to introduce Dr. Sargis today. He comes to us from UIC, the University of Illinois at Chicago. He received his BA in chemistry from Carleton College in Northfield, Minnesota, and then completed an MD PhD at Rush in Chicago, where he studied lipid and lipoprotein biochemistry under the mentorship of P.V. Subaya. And then he completed his internship and residency in internal medicine and fellowship in endocrinology at the University of Chicago, where he studied adipose physiology with Matthew Bradley, and that's where I became aware of his work several years ago. And then he was on the faculty at the University of Chicago before being recruited to UIC, where he's now an associate professor in the Division of Endocrinology, as well as the section chief of endocrinology at the VA, the Jesse Brown VA in Chicago. In addition to his research examining the impact of endocrine disrupting chemicals on metabolic physiology, Rob has an active clinical practice which is focused on patients with diabetes, lipid disorders, and other metabolic diseases. He is also the director of the Physician Scientist Development Program in the Department of Medicine at UIC. And his goal is to advance understanding of environmental pollutants as drivers of the current metabolic disease epidemic and health disparities. So his work spans the spectrum from cell-based and animal studies to clinical and population-based work. And he hopes that rigorous scientific inquiry examining the metabolic consequences of environmental contamination can serve as the foundation for public policy to mitigate the impact of pollution on human health. I'm very excited about your talk. The floor is yours.
Thank you very much, and thanks for the invitation to be here. It's been absolutely great talking to everybody. The visit's already been too short, but I'm looking forward to dinner and continuing the conversations. So I'm going to talk to you about sort of the spectrum of work that we do, sort of centered on endocrine disrupting chemicals, environmental health, and how it relates to metabolism. And at some point, I'm going to take a hard turn and talk a little bit about some of the newer things that we're doing that haven't been published yet, but I'm a little excited about. So by way of disclosures, I do some consulting work for CVS Health. I'm not responsible for the prior authorization forms you need to fill out, so you don't have to throw anything at me. I do some CME lecturing for the American Medical Forum, but none of this really impacts what I'm going to talk about today, which is really the mission of my lab, which is to understand how humans alter the environment and how those alterations impact human health. And I think the important thing to recognize is that at the base of this is really the ultimate goal, which is to translate that knowledge into action to protect metabolic health. So our whole goal is to take the science and translate it into policy ultimately. So what we're talking about today, just so that we're all on the same page, are these endocrine disrupting chemicals, which are defined by the Endocrine Society as exogenous chemicals or mixtures of chemicals that interfere with any aspect of hormone action. So traditionally, these have been thought of as chemicals that disrupt reproductive function, estrogen receptor signaling, androgen receptor signaling, thyroid hormone signaling. But over the course of probably the last 2 decades, we begin to recognize that these chemicals also play a role in the pathogenesis of obesity, diabetes, and affect pathways that regulate energy metabolism. Importantly, although the same chemicals are implicated, although I would say that these chemicals are also implicated in other diseases as well, including cancer, psychiatric disorders, neurologic disorders, which I'm not going to really discuss in any great detail, but I will talk a little bit about neurocognitive dysfunction. We'll move in that direction toward the end. So I don't need to tell this audience this, but let's make sure we're all on the same page, I suppose. And that is to say that diabetes is sort of the disease that is my greatest passion. It's defined phenotypically by elevations in blood glucose levels. But I think the important thing to really recognize about diabetes is that it really is a constellation of diseases. And I think think this is easiest to understand when we think about type 2 diabetes, which we typically associate with insulin resistance, obesity, physical inactivity, those types of things, and contrast that with type 1 diabetes, which makes up a smaller fraction of the disease, but is really more of an autoimmune beta cell dysfunction. But the important thing is, because we're talking about different diseases, it makes sense then that we may have different causes for these different presentations. And what we know epidemiologically is that we're seeing weird things happening, right? We're seeing disease in younger populations. We're seeing, when we're talking about type 2 diabetes, we're seeing type 1 diabetes in older populations. What this emphasizes to me is that something is happening. There's something changing. And the subtext of this talk is, well, is there something in the environment that may actually be driving these secular changes in diabetes? This is my favorite slide for understanding diabetes, and I will come back to this in a minute. But this is something that I frankly stole from Steve Kahn's paper in Nature in 2006, which basically summarizes clinical data showing the relationship between insulin sensitivity on the x-axis and insulin secretion on the y-axis. So as you all probably know, for those of us who have are normal glycemic, we exist on this curvilinear path in which insulin secretion is matched with insulin sensitivity to maintain blood glucose levels in a tight glycemic range. We find religion, we start exercising, we're riding our bike to work like Dr. Corbett, we're sleeping better, we're doing those things, we become progressively more insulin sensitive, and our body makes less insulin to maintain blood glucose levels in the normal range. The problem is that that's not where society is moving. In general, we're moving in the opposite direction. We're going to residency, we're eating donuts, we're not sleeping at all, we become progressively more insulin resistant. We slide up this curve, and in the short run, this isn't a problem. Our beta cells are able to compensate by making more insulin to maintain us in the euglycemic range. The problem is that this puts stress on the beta cells. They begin to fail. They initially fail during periods of peak demand, and you develop impaired glucose tolerance. And ultimately, the beta cells fail even in periods of fasting, and you develop frank type 2 diabetes. So at its core, what we're talking about with diabetes, type 2 diabetes, is this insulin resistance coupled with beta cell failure that ultimately leads to hyperglycemia. And I will return to this in a moment. So what drives this? So this is my real disclosure slide. And that is to say that I believe that genetics plays a role in all of this. I believe that diet has a role in the pathogenesis of diabetes. And I believe that physical inactivity also plays a role here. But the fact of the matter is that these factors alone fail to fully account for the magnitude and rapidity with which diabetes rates have increased in the U.S. and around the world. And this has raised questions about what other factors may actually be contributing to the rise in this pandemic. So this is a slide that I'm always a little embarrassed to show you, but this is from a paper that we put together back in 2011. And what you can see here, shown in red, is diabetes prevalence in the United States from the late 1950s until 2008. And plotted alongside that, meant to be provocative, is synthetic chemical production in the United States from 1938 to 2008. And you'll see this downturn in synthetic chemical production in 2008. This is not a trend. This is just the economic downturn from the Great Recession. And while obviously this is not causal data, it does raise the question as to whether these changes that we're making in the environment in terms of the synthetic chemicals that we're producing, releasing, and being exposed to, whether these things may have a causal role in the pathogenesis of type 2 diabetes, and in fact, all forms of diabetes. And so this has been generally the mission of my lab since we started. And the thing is, this becomes a really complex question because what we're talking about with these synthetic chemicals is a lot of different things, right? So this rise in synthetic chemical production includes chemicals that are used in food production. You can think of these as pesticides and food additives. Chemicals that are produced through industrial activity, including things like air pollutant, water pollutants, but also chemicals that we use in our own home, in personal care products, in home care products, cleaning supplies, flame retardants that are impregnated into our furniture or into our clothing. And then one of the things that really sticks in my craw is the fact that some of these chemicals are also used in medications and medical devices. And I'll return to this in a little bit later, but things like phthalates and bisphenols are in the medications we're prescribing to our patients, in the IV bags we're hanging in the NICU, and raising the question as to whether these may actually have detrimental effects that we don't fully appreciate. Now, linking chemicals to the pathogenesis of diabetes is actually quite old. So there's data in the animal literature from 1943 showing that exposure to alloxan, a chemical used in the production of a dye, could actually induce diabetes in the rat. And there's data from humans as early as the 1970s that exposure to the rodenticide Vacor could induce diabetic ketoacidosis in a subject who tried to commit suicide by consuming the rodenticide. So the premise that these chemicals may be altering beta cell function and thereby promoting the development of type 2 diabetes or type 1 diabetes is plausible and is borne out by the literature dating back decades. So when we look at the data as a whole, there's actually an increasing number of environmental contaminants that have been linked to disruptions in glucose homeostasis. These include chemicals that have been shown to alter insulin secretion, alter insulin action. They include both inorganic and organic compounds of natural and synthetic origin. And I'm going to talk about two that we've studied in the lab: talfluinide, which I'm going to try to argue induces a state of similar to type 2 diabetes, and arsenic, which induces a state a little bit more similar to type 1 diabetes. But what's important is when one looks at the consistency and diversity of the data, it does suggest that toxicant exposures represent novel diabetes risk factors. So the question is, why do we care? And the reason that we should care is that environments are modifiable. Unlike genetics, we can change our environment. We can alter exposures. And as such, this represents a tool that we can use to address this pandemic of disease that causes significant morbidity, mortality, and suffering amongst our patients and the population as a whole. So the first chemical that we started studying was this fungicide, talfluinide. So this is a phenylsulfonamide fungicide, historically used in Europe, also used in China, India, Australia, New Zealand. It is, in addition to being used on fruit crops, it's used as a booster biocide in marine paints. So these paints that are on the hulls of ships are usually copper-based paints that are impregnated with chemicals. Those impregnated chemicals are meant to prevent barnacles and algae and other things from adhering to the hulls of ships in order to improve hydrodynamics and reduce fuel consumption. But they're not covalently bound, so they leach into the marine ecosystem, can get incorporated into food. This chemical is not used in the United States, but it is allowed on imported crops. And the crops with the highest tolerance limit is hops. So just something to think about the next time you're having an IPA. And so we had done a bunch of studies looking at talfluinide. This is the study in which I did a Western looking at the effects of talfluinide on insulin signaling in adipose tissue. And Matthew took the gel and hit me over the head with it and said, "This is what you studied," because it had completely obliterated AKT phosphorylation in murine adipocytes. So we decided to take it into a mouse study. We studied at 65 parts per million, which is on the order of magnitude of what you would see if you only ate hops, but as principle. So what did we see? So what we saw with these mice, the only difference, same diet. These are C57 black 6 male mice treated with talfluinide in the diet at 65 parts per million. No changes in body weight over the first month, but then what we saw is that the mice treated with the fungicide gained more weight in the second 2 months of the study. That body weight was actually adiposity. There was no change in lean weight, but an increase in adiposity. And the mice developed glucose intolerance and insulin resistance. So a state that looked very much like type 2 diabetes. We also saw other interesting changes, including things like alterations in circadian rhythms. They consumed more food during the normal fasting light cycle, less during the dark cycle. And doing molecular studies, we believe that some of this is related to activation of glucocorticoid receptor signaling, which we were able to demonstrate both in vitro as well as in ex vivo in adipose tissue taken from these mice. But nobody cares about talfluinide, right? So it's like kind of this niche thing that I think I might be the only person that cares about. So part of the interest of the lab is to actually study things that matter for people's health, right, globally, right? And so that's what got us to study arsenic. So arsenic, and and I still have some regrets about this because it turns out that arsenic is this like really complex thing. So it's not like one thing. It's this complex metalloid that exists as in various forms in nature, including different oxidation states, inorganic forms, organic forms. But what's important is it contaminates the drinking water of somewhere around 100 to 150 million people around the globe. And in some cases, that exposure is significantly high. While it's a natural contaminant, it also has been used historically as a pesticide, with some arsenicals used in pigs and poultry. It's also found in various agricultural crops. So traditionally, you think about organic arsenic in seafood, but inorganic arsenic in rice. So rice is actually really good at accumulating heavy metals, toxic metals. And it turns out that brown rice is worse than white rice in terms of its accumulation of heavy metals because it gets incorporated into the husk. In an era of climate change, it's important to recognize that exposure is also increased by things like forest fires, and it is enriched in coal ash, which is one of the waste products of coal consumption. And then, as somebody who works at the VA, I think we often think about things like Agent Orange, but Agent Orange was one pesticide of many used in Vietnam, and Agent Blue was actually a mixture of arsenical pesticides that were sprayed in Vietnam. And it was something like at least 4.3 million liters of this was sprayed in Vietnam, such that there are areas in Vietnam that have high enough arsenic levels that put them on par with places like Bangladesh, which has been called by the World Health Organization the world's greatest poisoning due to arsenic contamination of well water. So what do we do? So in this study, what we did is we studied the impact of arsenic as sodium arsenite on glucose homeostasis in C57 black 6 male mice exposed to their drinking water. And what this shows you is that we got arsenic into the mice, so this is arsenic in their livers, not a surprise. The important point I want to say here, though, is that the arsenic levels in the liver of these mice were comparable to levels seen in highly exposed humans in Bangladesh. So we think that the internal levels of these are comparable to those who have been highly exposed. In contrast to talifluinid, we didn't see any changes in weight gain, but what we saw in these mice was a worsening of their glucose intolerance, no change in insulin sensitivity, but rather a reduction in insulin secretion, giving us the sense that this chemical, unlike talifluinid, created something that I would say is more type 1 diabetes-like. Not in the autoimmune aspects of the disease, but in the beta cell sort of focused aspects of the disease. So this fits all within a broader context of literature from others that have shown that a variety of different toxicants have been shown to alter both insulin sensitivity and beta cell insulin secretion. And some chemicals actually do both. And what's both awesome and scary at the same time is that the spectrum of evidence spans cells, tissues, animals, epidemiologic studies, and there have even been clinical studies in which people have been exposed to things like bisphenol A, showing disruptions in glucose homeostasis. So the argument, the other thing I would add is that these studies have improved in their mechanistic quality to the point where we're able to identify disruptions in signaling pathways that we know are related to disease development. So in total, what this suggests is when we start to think about the fact that each of us are exposed to somewhere between 2,000 and 3,000 unique chemicals of the 85,000 plus that are in consumer products, it's not hard to imagine that a little hit on insulin sensitivity and a little hit on beta cell function in some combination, coupled with genetics, coupled with lifestyle, may potentiate the development of metabolic deterioration. And so that's the argument that I would make, and I think that that's the argument that the data is making as we start to consider this in its totality. And again, it may not be one thing because each of us is exposed to many. So again, this could be something that we need to think about it as death by 1,000 cuts. So there's a lot of interest in mixture analyses as part of this. So so that's the mouse stuff we do, and no one is really honestly interested in mouse diabetes. Like, we study it because we're interested in what it means for human health, and as an endocrinologist, that's what I care about, right? So we were excuse me, lucky enough to hook up with Craig Hannon Dennis, who's at the University of Texas Health Science Center at Houston, who is a preeminent diabetes geneticist, epidemiologist, who's been doing work in Starr County, Texas for 40 years. And one of the good things about studying metals is you measure metals in urine, and everybody stores urine and doesn't know what to do with it or hasn't. So there's a lot of metal, there's a lot of potential for metal analyses in people's freezers. So we reached out to Craig to talk about this because Starr County, which can be seen here as the third county up along the Rio Grande River, is also corresponds to a hotspot of arsenic exposure based on USGS data. So we're like, well, can we do this? Can we start studying this in a human population in which this is probably particularly important? And we were able to do that. And this is a good place to be studying this because Starr County is somewhat of a unique place from a diabetes standpoint. So it's essentially 100% Mexican American. This is a population that we know has high risk of diabetes and is relatively understudied. It has an astronomically high poverty rate, but the rate of diabetes in Starr County, Texas, blows my mind. So the lifetime risk of developing diabetes if you're born in Starr County is 70%, which is part of the reason that they've been studying it from a genetic standpoint for a long time. So we hooked up with Craig to basically start asking questions about whether environmental factors may be contributing to the disease burden in Starr County. So this, I'm not an epidemiologist, so please don't ask me about the math. But we try to put a team together to kind of study this. So working with Maria Argos, who's an epidemiologist in our School of Public Health, and my MSTP student, Maggie Weiss, who's from Wisconsin, by the way, we started this project looking at the association between urinary metals and glycemic traits among the Starr County cohort. And this sort of gives you an idea of how that analysis was done. Specifically, this is our initial study, so we have an ongoing longitudinal study right now, but this is our first cross-sectional analysis to give you a sense of what types of things we found. So what we found was that arsenic exposure was associated with alterations in insulin sensitivity and beta cell function using steady-state measures of HOMA-B, HOMA-IR, and then HOMA-S, which is this thing that Craig uses, which is essentially a calculation that's very similar to HOMA-IR. It's basically the inverse of HOMA-IR. We also showed that copper was associated with alterations in beta cell function, and then molybdenum was associated with changes in insulin resistance. But what was weird is that the exposure to arsenic was, while associated with reductions in HOMA-B, was also associated with reductions in HOMA-IR. But as I told you before, this relationship or this role of arsenic is hard to decipher because it's complicated. It's multiple things, right? So it's not just one thing. So what we did is we dug in a little deeper to try to understand, well, what is it about arsenic, or what form of arsenic is actually associated with these changes, and then what does that mean? Because there are genetic determinants of arsenic metabolism. So first, I would just say that when we honed in on arsenic, we can show that total arsenic in the urine of these subjects, it's about 440 subjects, I think, was associated with a higher risk of diabetes. So again, cross-sectional, so we can't infer too much about causality here, but going from the lowest quartile to the highest quartile is about a 5.6-fold increase in diabetes risk for arsenic, which is pretty good with a positive trend there. Or I would say it's a negative trend for health, positive trend for the association. But then what we did is we dug into what species of arsenic might be related. So this is basically the way that arsenic gets metabolized, which is really kind of interesting in its own right. So there's an enzyme that metabolizes. We have an enzyme that metabolizes arsenic, which sort of speaks to perhaps the importance of arsenic over evolutionary history, but raises some important questions about why. So inorganic arsenic is converted to a monomethylated form of arsenic, which is converted to the dimethylated form of arsenic. That last conversion from monomethylated to dimethylated is the rate-limiting step. And what we found was that when we looked at HOMA-IR among 414 non-diabetic participants, so taking out those with diabetes from this analysis, that the higher the MMA you had, the lower your insulin resistance. And when we use the provocative measure of insulin sensitivity, the Matsuda index, which we had data on for 257 subjects, we saw the similar trend. Those folks who had higher MMA had lower insulin resistance. And so this is a really interesting phenomenon because it sort of suggests that having more MMA might actually be helpful. That it's the conversion of MMA to DMA that may actually be detrimental in terms of overall metabolic health. And what's especially bizarre about this is that MMA is thought to be the more toxic compound. And if you look at which forms of arsenic are associated with cancer, it is the MMA that is associated with cancer. So we end up with this interesting dichotomy when we start to think about arsenic metabolism and risk, where cancer risk may be dissociated from metabolic disease risk based on differential effects of either the dimethylated form from the monomethylated form. And again, this is all cross-sectional analyses, and we'll see how this bears out in the longitudinal analyses that are forthcoming. And this basically just summarizes that. Okay, so as I said before, we're interested in environments more generally. So not only are we people exposed to arsenic, but as I mentioned before, people are exposed to all sorts of different chemicals. And we know that diabetes rates vary dramatically across the country, and we know that environmental quality varies dramatically across the country. So one of the questions we wanted to ask is, well, okay, that's all well and good for one chemical, and we can study one chemical in an animal, or maybe we can study a couple chemicals in cells. But what about environmental quality as a whole? What about the summation of these effects across different domains? And so, and this is important because we know that environments often vary in the same direction, right? So what I mean by this is that they're often, if you live in a good environment, it's often good for many reasons, right? You have clean air, you have clean water, you have housing security, you don't have food insecurity, right? But similarly, environments can be deleterious in multiple ways simultaneously as well, right? There can be polluted air, polluted water, those types of things. So the Environmental Protection Agency developed a measure that looks at environment more broadly, and they call this the Environmental Quality Index. So the Environmental Quality Index is a summary variable that takes into account 5 domains of environmental health. So this includes air domain, water domain, land domain, built environment, and socio-demographic factors. And each of these individual domains is made up of multiple variables that constitute that domain. So we were interested in knowing, well, if you look across, and this is, I should say, this is data that's available at the county level, right? So, okay, so we can talk about how much that sucks. It's not ideal. Counties vary a lot depending on where you live, but this is what we have available. So the question was, well, how does environmental quality relate to things like diabetes control, diabetes prevalence, and we published now on both of these. So this is again a collaboration with Jyotsna Jagai, who is at UIC, now is at the University of Chicago. And what we did is we looked at the associations between the Environmental Quality Index and its subdomains, and county level estimates of diabetes control. So these are estimates that are based on Burfus data and NHANES data that have been developed by the Institute for Health Metrics and Evaluation at the University of Washington. It's all publicly available stuff that they were very willing to share with us. So we did these mixed effect random intercept linear regression models, and then we corrected for county level data on obesity and physical. And I understand that, again, this is not individual level data, but it gives us a sense sort of what types of phenomena we may be seeing. And there's complete data for 3,100 counties in the U.S. So what we saw with this data was that when one goes from the most healthy environment to the least healthy environment, there was a decrease in diabetes control. Those people with diabetes who had an A1C in the acceptable range. And when we look across the various domains, we saw similar decreasing trends for water, for land, for the built environment, and then what we would all suspect, which is that the socio-demographic factors are powerful drivers of diabetes control. The worse worse the sociodemographic factors in that county, the less likely people's diabetes was to be controlled. Again, showing that when we start to look at how these things interact, they interact in a way that suggests that environments may matter. Okay, so this is like, I don't know, maybe just because I grew up in Chicago, like I have this like interest in urban planning for some reason. Like, how do we build cities? How do we build environments that are actually healthy, right? So what we did, and I'm not sure this was a good idea because we haven't published it yet, but we haven't tried it yet, so that is not a bad idea yet. But I told you that the EQI data was made of 5 domains, and those 5 domains were made of multiple variables. So we said, okay, well, what about the other variables? Can we just go down to individual level variables? So what we did is we looked at urban areas in the United States, so just those urban counties, and basically looked to say, okay, what about air pollution? And then factors related to transportation, and how did those relate to diabetes prevalence and control? And John probably remembers I don't know if you call this when you're in Chicago, but this is the CTA bus. We used to call it the green limousine when we'd go to school. Sorry. So that was just a random Chicago aside. So anyway, what we found was that higher levels of particulate matter, 10 PM10, and higher NO2 levels were associated with higher prevalence of diabetes. And then when we looked at transportation factors, and I don't think anyone was surprised by this, but the worse public transportation use or the less transportation use associated with more diabetes, right, and less control. And walkability was associated, more walkability associated with less diabetes, right? So here, like, we should be thinking about this as we build cities. So as someone who lives in the suburb and spends an absurd amount of time in the car, like, that's terrible, right? And we're paying the price for that. So this is just meant to sort of get at that. And we want to do more data. We want to do more data. That doesn't even make any sense. We want to do more analyses with data at higher spatial resolution to really dig into this. But again, it all sort of points in a direction that suggests that we're neglecting environmental factors as potential drivers of metabolic disease risk. And here's a way we might be able to make it better. Okay. So one thing that sort of like is really stuck in my craw and like really drives me crazy and is really a passion for our work is that diabetes is a health justice issue, right? Like it is at its core a disease of injustice. And this is just the data, that compared to non-Hispanic whites, Hispanic here, non-Hispanic blacks, and American Indians all have higher rates of diabetes. It's sort of tragic because we've known this for a long time, and these disparities continue to, if anything, grow and not get better. And it's not just disease sort of prevalence, but we know that diabetes mortality is higher. So if you're African American living in the United States, your diabetes mortality rate is 2 times higher. So not only prevalence of disease, but the serious outcomes that manifest from it are disproportionately burdening certain communities. So obviously, there are a lot of factors that go into this: poor healthcare, economic barriers, education, insecurity in all its forms. But one of the things that we were interested in is whether environmental injustice may contribute to this, right? So is it possible that differential exposure to environmental pollution, at least partially, drives the higher rates of diabetes and diabetes-related complications in these populations? So in part of the paper I'm most proud of, this is Daniel Ruiz, who's a grad student in my lab, basically looked at this. This was a massive effort. It required like a 16-page proposal to Diabetes Care to get them to approve this. But what we looked at were chemicals associated with diabetes in the basic science literature, for which there was prospective human data to show associations between exposure and diabetes in the human populations. And so what we found is, it's not rocket science, but I think it illustrates the point. But across 5 different chemical classes, we showed that disadvantaged communities have higher levels of exposure to chemicals linked to disease. So this includes polychlorinated biphenyls, which are higher in non-whites and African Americans, or organochlorine pesticides. You can see the barnyard animals saying the DDT is good for me song here in the upper right. So higher levels in non-whites, particularly higher levels in Mexican Americans who are disproportionately represented in agriculture, higher levels of air pollution in those in areas of higher poverty, African Americans, non-whites, Hispanics, and Latinos. A lot having to do with racist housing policies, like things like redlining. That was another cool thing about this study, is that we paired with a sociologist to talk about drivers of this. And then for non-persistent pollutants like phthalates and bisphenols, higher exposure among those with low income, as well as those with lower food security. And I think one of the things that kind of sticks in my craw a little bit, higher levels among those who receive emergency food assistance, which makes sense because what we give these folks are canned foods, packaged foods, those types of things that increase their exposure to these chemicals. So what I hope I've argued for you so far is that environments matter from a metabolic disease standpoint, and they matter at multiple levels, which suggests that we need to start incorporating thinking about the environment into how we approach public health. Okay, so I'm the bald guy in this. That's a brain. I've been afraid of the brain since medical school. It's really complicated. I don't understand the circuitry, and I don't understand how we ended up here. But now I'm going to tell you a little bit of a story that goes beyond sugar a little bit, but relates to metabolic disruption that has to do with where we are with things right now. Okay. So we did this study looking at the addition of arsenic to a high-fat diet in C57 black 6 mice. And what we expected to find is that the arsenic plus the high-fat diet would make things worse. And of course, that's not what we saw. What we saw shown here is that exposure to arsenic decreased fat mass by DEXA scan as well as at necropsy, right? And there's a lot of spread in this data. So what we thought was, well, how does this relate to arsenic levels? And as it turns out, that the fat in these mice is directly proportional in a nonlinear way to the level of arsenic in that they had in their livers, right? So the more arsenic, the less fat they had. So this was something that was really interesting to us, and we kind of put it on the back burner for a while. But it started to raise questions about, well, what is arsenic doing in terms of fat metabolism and fatty acid species and all of this kind of stuff? And so Lisa mentioned that I did my PhD with P.V. Sabaya, who's had this long interest in essential fatty acids. And so essential fatty acids are, these omega-3 fatty acids. They include docosahexaenoic acid, which is 22 carbons, 6 double bonds. Really important essential fatty acids that have anti-inflammatory, antioxidant, and neuroprotective effects. And what's important is that this DHA is preferentially enriched in neurons, despite the fact that they can't synthesize it. So they're dependent on delivery from elsewhere. And we know that consumption of higher levels of omega-3 fatty acids are associated with a lower risk of depression. And then when you experimentally deplete omega-3 fatty acids in the diet of rats, they develop depression-like symptoms. And we know that other prenatal stresses can induce reductions in DHA levels. And so coupled with that is evidence that arsenic, the chemical we've been studying maybe to my chagrin, I don't know, for a long time, accumulates in the brain and induces depression-like behaviors. And specifically, developmental arsenic exposure induces cognitive impairments, depression-like symptoms. It affects multiple pathways, including hippocampal neurogenesis, HPA axis dysregulation. And we know epidemiologically, arsenic has been linked to multiple neurocognitive disorders. So mood disorders, anxiety disorders, learning and memory problems, as well as behavioral problems. So one of the questions was, is it possible these are related to each other? And we got samples in the freezer. So what do we got? So what we were able to show is in the brains of our mice exposed to arsenic. So these are just C57 black 6 mice on a chow diet. Exposed to arsenic for 8 weeks. There's a marked reduction in DHA levels in the brain, and actually a compensatory increase in arachidonic acid in the brain, which is important because arachidonic acid is pro-inflammatory. We also showed here that when we expose pregnant mice to arsenic, the dams as well as the pups have lower levels of DHA. The dams have the higher levels of arachidonic acid. There's no change in arachidonic acid levels in the pups, but the pups do have an increase in TNF-alpha expression in the brain of these mice. So the question is, well, does this affect behavior, and is it possible that maybe repleting DHA levels might actually be able to reverse some of these effects, right? And so this is super exciting. Like, as a clinician, like, it's kind of depressing spending your life studying things about how toxicants make things worse. Like, what are we going to do about it? So here's an opportunity. Maybe we've got a phenomenon here that we can reverse. So the problem with this, though, is that DHA doesn't exactly cross the blood-brain barrier. So it gets incorporated into triglycerides, which get incorporated into lipoproteins that effectively do not efficiently cross the blood-brain barrier. And so this was data so this is my PhD mentor who's got the office next door to me, although he's never around for some reason. But anyway, so he's done these studies in which he's shown that if you give free DHA levels to the mice, you do not get a change in DHA levels in the brain. But if you use a different molecular species of DHA, specifically lysophosphatidylcholine conjugated DHA, it does cross the blood-brain barrier. And so the reason for this is that there's a specific transporter at the blood-brain barrier, MFSD2A, that is specifically transports lysophosphatidyl lipids across the blood-brain barrier. So lysophosphatidylcholine, lysophosphatidylserine transports both. So the idea here, the model that we were working with, is that arsenic exposure depletes DHA levels and causing a mishmash of things to happen that causes neurocognitive dysfunction. And that by giving LPC DHA, we may be able to reverse these effects. Whereas if we give DHA in a triglyceride form, it doesn't cross the blood-brain barrier and is not able to accomplish this effect. So this is the model that we used. So basically, we exposed we exposed dams from 2 weeks prior to gestation through gestational lactation, and then so this is actually postnatal day 28, and then they get randomized to a diet, either control diet or LPC diet, DHA diet, or TGDHA diet. Now, our primary outcome was to look at the pups, but as an exploratory outcome, we said, what the heck, we got the dams. We know pregnancy is a period of vulnerability for the mom, so let's look at the dams as well. So what do we see? So no change in litter size, no change in sex ratio. I should mention that arsenic exposure here is super low, 500 parts per billion. So that's more than what you're going to see for the World Health Organization standard for human water consumption. But mice are really efficient at metabolizing arsenic. So this is actually a pretty reasonable dose for a mouse experiment. There's a slight increase in weaning weight, only driven by the number of mice we had, and it was both males and females. Female data shown here. So what did we see? So actually, we did see some changes in maternal behavior. So I've never done this before, and so this is why I'm telling you I'm afraid of the brain. So my interpretation of these behavioral studies should all be taken with a grain of salt, which is why everything says preliminary and unpublished on it. But we did see changes in maternal behavior. So they explored less in a novel object recognition test. There's some problems with that, but we can talk about it later. And on top of it, they had an increased percent freezing time during contextual fear conditioning, which could be, among other things, a potential model of post-traumatic stress disorder. So this arsenic exposure seemed to exert some negative effects on mom. We did not reverse mom, so this is something that we're interested in, because obviously these changes that happen in terms and the impact of things like postpartum depression, postpartum psychosis are pretty devastating for families, and if we could potentially do something about it, it would be great. So here's the data for the pups. So what's shown here is acute fear conditioning. So this is an effect that we only observed in females. So what acute fear conditioning does is you expose the pups to light, sound, shock, and then you expose them to light and sound, and you measure the amount of time that they freeze. And the idea is if they remember they got shocked, they will freeze for longer. Okay? And so what happens here is that when you expose the mice to arsenic, they remember the shock less, and so they freeze less, and this effect is reversed by the LPC DHA treatment, but not by the triglyceride form of DHA, which is pretty cool. And then what we did is contextual fear conditioning. So you do these tests under different contexts. So instead, what we do is we take the mice into the context in which they were shocked, and try to understand whether they recognize that they're in this context of being shocked and freeze. And what we can see when you take all the data as a whole is that the pups exposed to arsenic remember less, so they freeze less. And this effect is potently reversed by LPC-DHA, partially reversed by TG-DHA. And again, so this is obviously complicated because the arsenic is undoubtedly doing multiple things to these mice, but it does suggest that this nutraceutical intervention may be restoring DHA levels in normal neurocognitive function, or something like that in these mice. So a ton more work to do, but I just wanted to share some unpublished stuff with you because I don't know, it's kind of exciting if I have any idea what I'm doing, but I think it's got some promise. Okay, so now the question is, okay, so what I think I've shown you is that exposure to toxicants can disrupt metabolism. So then the thing becomes, well, that's all well and good. So what the hell are we going to do about it, right? So the question for all of us is, how do we translate this into action? And I think that this is something that's a bit in evolution, but is sort of kind of at the core of a lot of what I spend my time on. So obviously, the goal is to translate this knowledge to action, and we have the capacity to do this at multiple levels. So this includes individual level, local level, national level, and globally. And so we've written a little bit about this. So how do you reduce your exposures? How do we do that? So we've tried to provide some scholarship on this. We've also worked with Sabina Sheikh at the University of Chicago to understand how current federal environmental policy handles endocrine disrupting chemicals and their potential relationship with diabetes. The answer is it doesn't. So creating this important policy deficit that needs to be filled. And I also think that there's a role for us as clinicians, given the fact that we know that some of these endocrine disrupting chemicals are in medications and IV bags and these things. And what's interesting about that is that means that we're the vectors of exposure. We are the mosquitoes transmitting malaria, if you will, right? So this, I think, imposes really important ethical import to us, right? I mean, we have ethical obligations to our patients to make sure that we're maximizing benefit and minimizing risk. And I would suggest that we never talk about this and we never inform patients. So from the standpoint of patient autonomy and non-maleficence, we need to start thinking about what we're exposing our patients to. And so we've written a little bit about the ethical implications of all of this in this paper that was published in JCM not too long. Okay. The other thing is, how do we get more people involved and engaged? And I think I'm specifically speaking to the trainees here. And it's really exciting that over the course, I don't know when this happened. So in the last 6 months, we now have an endocrine society special interest group devoted to endocrine disrupting chemicals. So I'm on the advisory board for this. There's the effort of Tom Zeller and Andrea Gore and Jerry Heindel, people who have really been leaders in this field for a long time. And so if you're interested, we've got events planned. It looks like October, we're going to have an event on obesogens. If you're interested, sign up. The one thing that they keep telling me is that if you want to sign up, you got to be a member of the Endocrine Society. So just keep that in mind. But there are ways for you to get engaged in this on multiple levels if this is interesting. And the Endocrine Society is really the home for all of this. So with that, I think I'll close there and just thank all the cool people that I get a chance to work with. They do all the work, and so I'm super grateful for funders. If you want to hear me rant on Twitter about local environmental issues, which I can talk about, there I am. But thank you again for the invitation. This is so invigorating. I'm so excited to be here. So, all right, now tell me how I'm wrong, I guess, or yell at me or something. I don't know. But thanks so much.
Thank you for this excellent talk. I will open it for discussion or questions.
Did Mr. Ryan in the green limousine?
So what?
Did Mr. Ryan in the green limousine?
I do. I can still ride in the green limousine though sometimes. They're not green anymore, but that's the only thing. And there's air conditioning now, which is really only for people who are soft.
I have a question.
Yeah, please.
So in your arsenic brain story, did I miss any link to the metabolic
So, so some of that we haven't really measured because we were so focused on doing the behavioral testing and getting that done. There are slight changes though in those weaning weights that suggest that there may be changes. So I think a lot of that data is still to come. There's some published data looking at similar doses of arsenic that have suggested that developmental exposure to arsenic at that level increases the risk of obesity later. There's studies that suggest that, and this is, I think, interesting because it relates to stuff that we've talked about, that it may be due to reductions in energy expenditure due to alterations in sympathetic activity in brown adipose tissue, specifically from the developmental exposure window, which is obviously different than what we did in the adult mice. So I think there's more data for us to get there. We're so focused on like trying to understand like these behavioral tests that we had never done. I just showed you some of them. We did a bunch of other things. So I want to move in that direction. We didn't see anything hugely overt in terms of glucose, fasting glucose levels or insulin, but probably more to do in terms of sophisticated studies. And then the other thing I would add is, we did these behavioral studies, but it's still open a little bit as to like where or what sort of domains the arsenic is most likely to affect. So we're doing some cool stuff now, looking at imaging mass spec to really understand where the DHA levels are in the brain and how the arsenic may be differentially affected in the brain, which may hone into certain areas. And where I'm going with that is, one of the areas of interest is the hypothalamus. So the evidence suggests that the hypothalamus and the pituitary are regions in which arsenic does get enriched when mice get exposed. And last week, I don't have the data yet, so I don't know if this is going to shake out, but last week we sent some brain sections to the synchrotron at Oregon where they can do X-ray fluorescence spectroscopy. And what we're trying to hone in on is whether we can see that in our own mice, whether arsenic is rich in the hypothalamus, hippocampus, or amygdala, which are the regions we're thinking of. If there is enrichment in the hypothalamus in particular, I think we'll do more provocative metabolic testing that kind of really dig into what might be going on. Yeah.
So EPAs are consequential gases. I think they're also important for
Yeah.
Did you have to look at that?
Yeah. So the thing with EPA is that its levels are much lower than DHA, so it's a smaller increase. The nice thing about EPA though is if you increase EPA delivery to the brain, it's converted to DHA, so you can actually kill two birds with one stone. So one of the problems with this study is that LPC DHA is like absurdly expensive. So what you can do is you can use light-based snake venom treated krill oil, and then you get a mixture of the two. So I think when we think about scalability and moving on to sort of next phases, that's going to be probably part of it. So we'll probably look at a dual effect. In the initial pilot stuff, we wanted to really try to keep it as clean as possible, but EPA is also important. Just a smaller fraction. That's all.
So you got being a neurobiologist, the changes you see in freeze time and all that stuff, how do those compare to stimuli that really drive neural effects?
Yeah.
Like so maybe lead, for example. I don't know if that's a good choice of poison that.
Yeah. So this is an interesting thing because because as I said, I was afraid of the brain and not a neurobiologist, but I presented this at the Steroids in the Brain conference in Brackenridge in March, and they seemed to be somewhat impressed with like the level of the effect that we were seeing. So I think we might be onto something. One of the questions we have is it unique to arsenic, or is it basically any pro-oxidant metal going to do something similar, right? And so we got a pilot grant specifically to look at hexavalent chromium. So hexavalent chromium, you probably know because it's the Aaron Brockovich chemical. So it's a known carcinogen. But there was an RFA, our center coupled with the Gulf Coast Center for Precision and Environmental Health put together an RFA specifically looking at military-associated exposures. And folks in the military have differential exposure to this because it's used in paints on war, on planes and ammunition, and so they get exposed. So we're actually doing studies now to see if some of the behavioral and molecular changes, metabolic changes we're seeing, are similar for hexavalent chromium and arsenic. But yeah, I mean, lead probably does the same thing. A bunch of things probably do the same thing. And then the thing you get into is like, well, okay, it's a nutraceutical, like it's doing all these things. universal for some things that are permanent are still helpful. So, but I think there's an open question about that. And then we have plenty of questions about mechanism.
I know, sir. I'm just curious.
Yeah.
So, have you ever done this in a different mouse strain than box 6?
Yeah, that's a really good question. So, first of all, it would be great if we didn't have to use mice at all for arsenic, because if we're looking at behavior, mice are terrible. Like you see all the variants in the data, their behavior is not that sophisticated. So the usual question is, well, why don't you study it in rats? I'm like, well, yeah, we'd love to, but our rats metabolize it differently. But your point is well taken. We probably, I mean, if we want to extrapolate this stuff, really look at outbred.
Or do mouse metabolize well?
So what rats do is that they put the arsenic in their red blood cells. So they have this alternative mechanism of detoxification that's not applicable to humans. So that's generally why it's not studied. But we should be looking at outbred strains for sure. And then one of the other things that this just came up the other day, but
sensitive strain. I think it's something that is highly insulin sensitive to.
It's a great suggestion. We should probably do that. I think we use them because we got them mostly, and we can genetically manipulate them.
Yeah.
Yeah.
So then another question is, so you feed the mothers, they have pups, you look at the pups, have you ever looked at treating the pups with arsenic to see if they're super sensitive because of whatever genetic change?
Yeah, I would say it's on the list. It's like the 2-hit thing, right?
Yeah.
The other thing we're interested in is, and you can tell me if I'm crazy here too, is like, what about other secondary neurologic stressors, right? So the thing that jumps out is both in terms of like our veteran population, but also the pandemic is like, what about priming social isolation, right? Does this potentiate the adverse effects of social isolation or dietary changes or whatever? I mean, you could think about a whole bunch of things. But what's interesting about your hypothesis a little bit is, does it build resistance somehow too, right? Like, could they be more resistant if they've been pre-exposed, right? Yeah. So these are all, like, I think, great questions that we have to ask.
Last one.
Please.
You use the word metabolism, and you talk great lengths about really crude measures of metabolism. Have you looked at metabolism in any tissue? We're looking at glucose oxidation to CO2, but does arsenic change the rates of oxidation? Because your whole phenotype could be a 10% reduction in insulin secretion or a 10% reduction in insulin action.
Yeah.
And a 10% reduction in glycolysis would give you a 10% decrease in insulin secretion.
Yeah, so we have not done those type of really sophisticated metabolic studies, and I think that's my fault. Probably we need to move more in those directions. So mostly we do sort of more of the crude physiology type of stuff.
Pretty good inhibitor.
It does a lot. I mean, this is the problem with studying these toxicants, is they're not drugs, right? So you've got multiple things that are probably going on, and this gets at that whole idea of mixtures too, which is it's death by 1,000 cuts, a little bit here, as you're saying, a little bit there, and sort of having these effects in different tissues doing different things. So no, it's completely fair, and that's what I think makes it challenging. And that's why the treatment in the end is don't get exposed to arsenic in the first place, as opposed to a specific drug to reverse the effect. Yeah. Where there are no forest fires and no one burns coal, and yeah, we'll have to figure that out. Yeah, please, Daisy.
So I was scanning your list looking for something that I've been very interested in, which is ACO. Of course, this is a conservation product, but also found in environmental fluids.
Yeah.
Exhaust, also in beer,
Yeah.
and donuts and bottled water. Have you ever thought about looking at
No, but we should talk about it at dinner. Yeah, I have not thought about it at all. But I'm interested in it at all. I mean, you can probably tell that. Like, but definitely, like, I'm open to any ideas here. I kind of, it's interesting because there's like the metal part of it, and then there's the sort of the organic part of it also. We sort of very rarely look at the combination of the two also. So I think there's some really interesting questions there, and then how we put these together. And if you look at something like acrolein, my guess is that probably exposure is pretty widespread, and so that again makes it probably more interesting from a public health standpoint. But I haven't to date.
especially in urban planning?
Oh yeah, for sure. Yeah. I love cities. We should just build them better, and we shouldn't be exposing ourselves to all this nonsense. Yeah.
Question about the first chemical that you mentioned, the toluene. You mentioned that in your experiments, it increased food intake during the life cycle, and so it suggests that maybe it was causing some kind of circadian disruption. Did you look at other markers of circadian rhythm?
No, we proposed doing that, and then the grant like completely crashed. Like, there was no interest in funding further work on that. But it fit. It made sense to us because it kind of fit with a circadian disruption induced by glucocorticoids receptor signaling. And it's interesting because it might be something that might be worth going back to now that we're doing these behavioral studies, where we can do we have a room that is still just ours, and I'm not sure how this has happened, but we can reverse the light dark cycles. We could do behavioral testing at under different light cycles. So it would be something that would be interesting to look at. We kind of got away from it because the EU banned it, like around the time our studies were coming out, and so we wanted to move to something more interesting. But from like a toxicant that we have a better handle on its mechanism as proof of principle, it would be interesting to look at more circadian markers in those lights. But food intake was the inactivity, I think, were the things that we saw. So metabolic ages.
How long does it persist in the environment?
So tolerant has an octanol to water partition coefficient of 3.9, so it partitions the fat pretty well. So it probably gets degraded in aqueous environments okay, but we would expect that in the mouse exposed that it would stick around for a bit.
Last question, is the seafood and fish significant source of this chemical?
So there were studies done in India that showed higher levels, but I think it was water. But you wouldn't predict it. So we know that seafood enriches other chemicals that are used for the same purpose. So I didn't show, we have a little bit of data on tributyltin. Tributyltin is like the classic obesogen, right, that everybody studies. It has transgenerational effects. That has been shown to exist in seafood. It induces imposex, intersex in mollusks. So it causes mollusk population collapse off the coast of France. So I would expect that it would be there. It just hasn't been measured very much is the problem. And then so that creates problems like scientifically as well, because then it's like, well, if it's not measured, and it makes it hard to make the argument to study it more. Right?
It's a very thought-provoking presentation, but I could probably talk to you about an hour about this. But focusing on the Mexican-American population in some Texas, there are a lot of complications in there. I mean, it's very good to know that there's a toxicant because it can unclench or it can bring to the surface some of the underlying genetic susceptibilities. Now, what we know about the Mexican population is that it has 10% inherited risk of toxicity. But it is in its environment. Simple environment has a changing diet. From the Mexico, United States, your risk goes to 20. If you go in part what you mentioned, low income, they go to fast food, low quality food, it goes up to 40. However, when we took our patients in Mexico, everybody is considered to be pre-diabetic. Without doing tests, you're pre-diabetic because you're Mexican. So you treat them as you're preventing the development of disease, and you do get people to go all their lives without developing even abnormal fasting glucose. But here's where some of your data might add some insights: is the fact that if you go to two environments, one where you have 5 species of hogs, it's going to be pretty clean. If you go to some, let's say downtown Mexico City, the environment is so different. Somebody might survive all their lives without any problems where the hogs live, but not in Mexico City, downtown LA, and so forth. But the problem is there's so many other environmental factors, including simple things like traditional diets. Losing the traditional diet just destroys your health.
No, it's all a fair point, and that's why I envision this as I should have put the slide in, but I think the slide only makes sense to me. So if you think about a reaction coordinate in chemistry in which you go from reactants to products and you have an activation energy, you extend that analogy to the development of disease and you go from health to disease, there's resistance to that development. And that resistance, that transition state is brought down by numerous factors, right? It's brought down by genetics. It's brought down by physical inactivity. It's brought down by bad diet. And so what I would argue is that, in certain instances, one thing could bring it all the way down, right? You've got a monogenic form of diabetes. Your transition state energy is super low, right? But the reality is what you're saying. The reality is it's probably death by 1,000 cuts. It's a little bit of things that potentiate that underlying predisposition. And so while the activation energy might be a little bit lower because of the genetics, maybe the environment is what sort of gets you to the tipping point. So the great thing about working with Craig is that he's been in this community for so long that they have such a good relationship with the community that they're gathering data on things like diet. And we're actually going to do gene by environment interactions. So we have some data already showing that some of the SNPs related to arsenic methyltransferase do seem to be related to some of the traits. But I agree. I mean, but then again, this is the thing with complex traits is like you want to address the addressable. And so your point is well